last stage of protein synthesis is translation, and this is where the mature mRNA molecule is actually used to make a polypeptide chain. Before we look at translation itself, we need to look at how the DNA controls protein synthesis. The sequence of bases on the DNA determines the sequence of bases on the messenger RNA, or the mRNAs. In turn, the sequence of bases on the mRNAs determine the sequence of amino acids that are joined together, and this sequence of amino acids determine the polypeptide that is formed, and the polypeptides will then form a protein. So how does the sequence of bases on mRNAs affect the sequence of amino acids in a polypeptide? Consecutive triplet bases or codons on mRNAs correspond to an amino acid. So for example, the codon UAU would correspond to the amino acid tyrosine, and this correspondence is called the genetic code. But why does it have to be in triplets? There are 20 amino acids in total in the human body, so one base cannot be designated to one amino acid since we'd only have 4 possible combinations, thus 4 amino acids, which is not sufficient. The same case is applied for 2 bases. We'd only have 4 times 4 or 16 possible combinations, which is also not sufficient. However, if we have 3 bases, this is perfect because we'd have 4 times 4 times 4, which is 64 possible combinations. Because the number of possible combinations is greater than the number of amino acids actually needed, you might be wondering, what is 64 codons used for? So 3 of those codons, UAA, UGA, and UAG, are stop codons, which signify the termination point for translation. The other 61 codons are used to code for amino acids. From this, you may infer that although one codon can only code for one amino acid, one amino acid can be coded by several codons. Now looking more closely at translation itself. Overall, this occurs when an mRNA is binded to a tRNA molecule inside the ribosomal structure. Looking more specifically into it, you will notice that the mRNA only provides a template for the polypeptide chain that is being formed. Therefore, it does not directly interact with the amino acid itself. However, the mRNA and the amino acid interact through the tRNA molecule. First, the ribosome would attach itself onto an initiation site on the mRNA, which is basically just a point where there's the codon AUG. This codon corresponds with the amino acid methionine, as specified here. This will help initiate the process of translation. However, the amino acid methionine is often removed from the polypeptide before polypeptide synthesis completes. The ribosome provides ribosomal RNAs, which are used to help bind the mRNA and the tRNA molecules together. And then afterwards, as you can see here, once the first tRNA molecule has finished its job, the second tRNA molecule will come in to bind to the mRNA on the next codon using complementary base pairing. And the ribosome also provides enzymes which help the formation of the amide bond between the 2 amino acids. So therefore, the amino acid on the first tRNA molecule is then moved here, and this is a growing polypeptide chain. And this sequence of events will continue, i.e. there will be more tRNAs coming, and the amino acid which they carry will then be added onto the polypeptide chain, and the previous tRNA molecule will be removed. This sequence of events, it will continue until a stop codon is met in the direction of 5' to 3'. I want to introduce the transfer RNA molecule in a little bit more depth because in the previous diagram, as seen here, it is demonstrated a little bit like a price tag, which it's really not. You will see here that the transfer RNA molecule actually looks a little bit like a clover leaf. It has an amino attachment site at the 3' end right here, the loop, T-loop, and the anticodon as introduced, and right above that there's an anticodon loop. And this is quite similar to the previous diagrams, so it just depicts the binding of the anticodon to the codon using complementary base pairing. Next, we look at post-translational modifications, and this is an important stage before a polypeptide chain can become a functional protein. So the polypeptide chain can either cleavage or split in order to form smaller proteins, or they can combine with other polypeptide chains to form bigger proteins, or there can be the addition of carbohydrate side chains to the polypeptide. That's all for the series of protein synthesis. Please like and subscribe for more molecular biology videos.